Welcome everyone, I'm Zaina Syed, I'm a third year medical student, and I'll be giving this session on the feed-fast cycle. Okay, so this is what we're discussing today. We'll start with a recap on hormones and energy sources. I really recommend going through the previous MOL lectures you guys took because it'll help you understand this lecture better. It includes a lot of basic stuff. And then we'll move on to the 3 phases of feed-fast cycle, starting with absorptive phase, post-absorptive phase, and then lastly, starvation. Okay. Now, Now, 3 main hormones involved in the feed-fast cycle. We have insulin, glucagon, and then we have counter-regulatory hormones. We'll discuss the last one later because it's not very important. The more important ones here are insulin and glucagon. Okay, can anyone tell me, I'm pretty sure you guys took this before, can anyone tell me any function of insulin? What does it do? Reduce blood sugar. Okay. Now, if we're trying to glucose storage, okay. Now, if we're trying to divide the functions, we could look at it as insulin, the effects it has on glucose, lipids, or proteins. Okay. Now, someone said glucose storage. Okay. Now, the 3 main effects insulin has on glucose. One, it increases glucose uptake by activating certain glucose transporters called GLUT4. We'll look into this later. And then it enhances glucose synthesis. Glycogen synthesis. And lastly, it decreases glucose production. Now, what are the 2 main processes that produce glucose? We have glycogenolysis, that's breakdown of glycogen, and then we have gluconeogenesis, that's making glucose from amino acids. Okay, then we can look into the effects of insulin on lipids. Does anyone know any? Okay, increase lipid uptake. Anything else? Activates an enzyme. Okay. Basically, okay. One, it increases glucose uptake in adipose tissue. Yes, this is done for lipid synthesis. And then we have increase in triglyceride synthesis. How does this happen? Insulin basically activates a certain enzyme called lipoprotein lipase. Now, what does this do? And if you guys remember, lipoprotein lipase basically breaks down the circulating forms of lipid carriers like chylomicrons. And when it breaks it down, it releases glycerol. Now, glycerol combines with fatty acids and it forms triacylglycerols. And lastly, there's a certain enzyme called hormone sensitive lipase. And from its name, lipase, it breaks down fats, right? Now, insulin, what it does, it dephosphorates this enzyme. So it reduces the activity and you have less fat breakdown. Lastly, proteins, you have increased amino acid uptake into the cell. So if amino acids go into the cell, they synthesize proteins, so enhances protein synthesis. And the next hormone, glucagon, does exactly the opposite of what insulin does. So when we said insulin increases glucose uptake, enhances glycogen synthesis, decreases glucose production, what do you think glucagon would do? What effect would it have on carbohydrates? Decreases what? Okay, increases glucose production, more glycogen lysis. Yes, it stimulates glycogenolysis, which is glycogen breakdown, and it promotes gluconeogenesis, glucose production. And what effect would it have on lipids? It stimulates fatty acid oxidation and ketone body formation. Lastly, for proteins, it increases amino acid uptake, similarly like insulin does. But here we need the amino acids for gluconeogenesis. Okay. Moving on, let's look into the major energy sources. We all know we have carbohydrates, lipids, and proteins. Okay, and carbohydrates is basically the main. It basically provides immediate energy. So we have glucose and the storage form of glucose, that's glycogen. So when you take in, when you eat something, now your organs, the first thing they use glycogen from the food. So it provides immediate energy. The second, when we have glucose depleted, what would the body do? It start using up lipids, triglycerides, fatty acids, and ketone bodies. We'll look into each of these separately later. And lipids are the largest energy reserve. So you have a lot of it used up. And lastly, the last energy resort would be protein. So once we're done with carbohydrates and lipids, now if we have nothing, we use proteins. Muscle proteins, we break down proteins. What does protein break down form? Amino acids. And amino acids is used in a process called gluconeogenesis, like we said, forming glucose. Okay. Is everyone clear with the basics? We can move on. Okay. Okay, now let's start with the first phase of the fast cycle, which is the well-fed stage. So basically, when you take in a meal, 0 to 3 hours after the meal, we have high glucose in the we have excess glucose in the blood, right? So we have high glucose amount. What would that do? It would cause the release of insulin from pancreatic B cells. Okay, so we'd have high insulin. Um, and the opposite would happen to glucagon. So you have high insulin and you have low glucagon. What would that do? That would increase the insulin-glucagon ratio. So when you have a high ratio, and basically the body is trying to activate stimulating the anabolic pathways. Anabolic meaning we have high glucose, so you want to store it, right? Yeah. Okay. So let's look into what happens to each organ in this phase. In the phase where we have high insulin released, and we all know what the effects of insulin, so we're basically just going to see how it affects each organ. And one thing to keep in mind before we move on, there are two organs that are insensitive to insulin, which are brain and red blood cells. So no matter what the levels of insulin is, it's not going to affect glucose, the production of energy by glucose. Okay, now let's start with brain. We said already they're insensitive to insulin. Now there are we said one of the functions of insulin is to cause glucose uptake, right? Since the brain is insensitive, we have certain glucose transporters, GLUT1 and GLUT3. These cause glucose to enter into the cell. Glucose basically here is a primary fuel for producing energy. Now, since we said the brain is insensitive to insulin, do you think GLUT1 and because we said before, if you guys remember, we said before, insulin activates GLUT4, right? It's another glucose transporter. So in this case, do you think GLUT1 and GLUT3 would be activated by insulin? No. Okay. Yes, this is all we have to remember for brain. And then let's move on to red blood cells. Okay. Red blood cells are also insensitive to insulin. Now, glucose, once inside the red blood cells, it undergoes anaerobic glycolysis to produce pyruvate. This process releases ATP. This is how RBCs get their glucose. Another important thing to remember for RBCs is that glucose is the only energy source they have. So whether you're in your feeding state or your fasting state or your starvation state, it's only going to be glucose. Okay. Okay. Now, after glucose gets converted to pyruvate, it gets converted to lactate, and then that goes to the liver. We'll look into that later. Okay. Now, what happens in the liver? Could anyone tell me what the major energy source would be for the liver? Now, which one of the macro fats, protein? And if you guys remember, in this state, in this phase, we have glucose. We have high levels of glucose, we have high levels of insulin. Yes, it would be glucose, but one of the main energy sources would be oxidation of excess amino acids. Now, what the body does, when you have amino acids, it gets converted to pyruvate, gets converted to acetyl-CoA, that eventually gets entered the Krebs cycle and forms ATP. But this doesn't mean that it doesn't use glucose. It does use glucose as its energy source, but the main one would be oxidation of amino acids, okay? So once that's used up, it moves on to glucose being used as an energy source. This is basically how it works. Amino acids get converted to pyruvate, and then acetyl-CoA enters the Krebs cycle, forms ATP. That's what oxidation of amino acid means. Okay. Okay, now that we know the liver uses amino acids first as its energy source, it also undergoes a few processes. Now we know we have high levels of glucose, right? So what does the liver do? It tries to store it and stores it in the form of glycogen. So you have glycogen synthesis. And then we also have increased fat synthesis. But this only happens after the glycogen stores are full. So you have glucose that comes in, gets built up, glycogen starts forming, and then when you have enough glycogen stores, um, excess glucose gets converted into fatty acids. Okay, this is how it happens. Glucose is converted to glycogen, and then in fat synthesis, glucose turns to pyruvate, to acetyl-CoA, and then that forms fatty acids. And then we have glycerol P here. Glycerol basically comes from something called chylomicrons. If you remember, chylomicrons is basically lipids being carried, dietary lipids being carried. So that gives the liver glycerol, and then that combines with fatty acids to form fats. Okay. Muscle's main energy source would be glucose, because we have high levels of glucose. And it would do three things. One, glucose would get into the cell through specific glucose transporters called GLUT4. Okay, like we said before, these are insulin dependent, and we know we have high insulin levels in the feeding stage. Next, it causes glycogen synthesis, and then lastly, it also causes increased protein synthesis. Again, if you remember, insulin causes amino acid uptake into the muscle cells, right? So if you have more amino acids, they synthesize proteins. Lastly, adipose tissue would also would also use glucose as an energy source, and then it would be involved in increasing fat synthesis. And how does this happen? We have glucose. Glucose enters the cell. It gets converted to pyruvate, acetyl-CoA. Now, this whole process of it going to Krebs cycle, forming ATP, this is how the adipose tissue itself gets its energy. Any excess glucose gets converted to glycerol, and then that combines with fatty acids. These fatty acids, they come from VLDLs from the liver, and then fatty acids and glycerol combine to form fat. If you guys want to remember it, and just to understand, it's not very important, but if you remember, we said before in the liver that we're getting our fatty acids from glucose, and then we're getting our glycerol P from chylomicrons. And these combine to form fat. However, here in adipose tissue, we have glucose forming glycerol P, and then fatty acids that come from VLDLs to form fat. Okay? Now, this picture was in your slides, and you don't really need to know the details here. And all you need to know here is what metabolic pathways each organ carries out. Like we said, and like I summarized it before, muscles. Now we have glycogen synthesis in muscles. We have glycogen synthesis in the liver, fat synthesis in the liver, adipose tissue. And this is all you basically have to know. Okay. Okay. And we know what each one of these organs, their main energy fuel is. The brain, you have the glucose. Red blood cells, we have glucose. Liver, mainly we have oxidation of amino acids, and then we have glucose. And then muscles, adipose tissue, they're all glucose. The only different one here is the liver. Okay. Does anyone have any questions before I move on? Okay. Now, the second stage is the post-absorptive early fast stage. This happens 3 to 18 hours after a meal. So if you think about it, at this point, the glucose levels in your body are already low. If there's less glucose, there's less insulin released from pancreas cells and more glucagon. So there's low insulin, there's high glucagon, and the ratio would be low. This means, more of catabolic pathways get activated, and makes sense because we have low glucose. And so we need to break down more glycogen to form glucose. Okay. RBCs, like we said before, they're in synthesis of insulin, and we know that the main energy source for RBCs, or the only energy source actually for RBCs, is glucose. So whatever happens, whether we're in the feeding state, fasting state, it's only going to be glucose. Now the way the RBCs get the glucose might differ, because right now in the fasting stage, we have less glucose. So this glucose might come from other processes like gluconeogenesis, glycogen breakdown, lipolysis. Okay. Now, okay. In this stage, the glucose remains the primary fuel for brain. However, like I said for RBCs, where this glucose comes is different. Before we said the glucose comes from is dietary. But here we have it from, we get the glucose from lipid breaking down glycogen and lipid making it from gluconeogenesis. That's the only difference. Okay, let's move on to liver. Okay, now the major energy source for liver now would be fatty acids because we have low glucose. No, that's not an energy source anymore. And fatty acids, where do we get these fatty acids from? It will be from adipose tissue. It will break down fats by lipolysis and form fatty acids. These fatty acids would go to different organs. One of them is the liver and provide energy. Okay. Okay, now the two pathways that happen in the liver, one's glycogenolysis, which is breakdown of glycogen. Makes sense because we have low glucose and we need more, so we break down the glycogen stores. And once we use up the glycogen stores, we move on to gluconeogenesis, forming glucose from amino acids. Okay. Okay, this is basically what gluconeogenesis is. I'm pretty sure you guys know the process, but it's alanine amino acids that gets converted to pyruvate, then pyruvate combines with glycerol to form glucose, and then similarly, red blood cells, we said that they form lactate from pyruvate. This gets into the liver, gets converted to pyruvate, and eventually glucose forms. That's just the process of gluconeogenesis. Okay, now moving on to muscles. Now, muscles, also their primary energy source would be fatty acids. Where do we get these fatty acids again? We get it from adipose tissue breaking down fats. Now, okay, what does the muscle do? It increases protein breakdown. So we know protein breaking down, it forms amino acids. Why do we need these amino acids? We said before that the liver carries out gluconeogenesis, right? And where do we get the amino acids from for gluconeogenesis? We get it from the muscle. That means protein breakdown increases. This causes more amino acids, moves into the liver, and carries out gluconeogenesis. Okay. Now moving on to adipose tissue. Again, its main energy source is fatty acids. What does adipose tissue do? It causes lipolysis. How does this happen? There's a specific enzyme, we should mention it before, it's hormone sensitive lipase. From its name, lipase breaks down lipids. Okay, it breaks down triacylglycerol to fatty acids and glycerol. Now, these fatty acids, they could either be converted to acetyl-CoA, enter the Krebs cycle, and form ATP for the tissue itself, or it could go into the blood and to other organs, like we mentioned before, in the liver, muscle. They also use fatty acids as an energy source. Okay. Again, this diagram basically summarizes everything. This is just to show how integrated everything is. And you don't need to really know you don't really need to know the details here because I'm pretty sure you took it before, but for the sake of this lecture, and all you need to know is what metabolic pathways happen in each of these organs and what energy source each organ uses, each cell uses. Okay. Does anyone have any questions before we move on? Okay. Now, last stage is the prolonged fasting stage. This is starvation. This is 24 hours after you've had a meal. So you can imagine the glucose levels in the blood would be really, really low. What does that mean? We'd have very low insulin and will be very high glucagon released. And then you'd also have elevated counter-regulatory hormones. Now, there's not much a doctor said about these, but now all you need to know is that now if you can guess, starvation is basically when there's low glucose. So obviously you'd need more metabolic pathways like gluconeogenesis, lipolysis, glycogen breakdown, and all of these to happen. So these hormones would help in increasing them. Okay. Okay. Can anyone guess what would happen in RBCs at this stage? Yes, like we said, nothing changes. It is glucose dependent, so glucose would get converted to pyruvate, and then this process would release ATP. Okay, however, in the brain, glucose was the primary fuel, right? This changes, and ketones instead becomes the major energy source for the brain. How do we get these ketones? We'll see it later. Okay. Now, in the liver, the fatty acids, they remain the main energy source. Again, we said fatty acids, we get it from adipose tissue, breaking down of fats, by breaking down of fats. Okay. Now, these are the three things that go on in the liver. One, gluconeogenesis. Now, we know where we have very low glucose and then glycogen stores are depleted, so we won't have glycogen breakdown. We start with gluconeogenesis, okay? And then we'd also have ketone body production. How does this happen? It's basically fatty acids from adipose tissue gets converted to acetyl-CoA, and this forms ketone bodies. Now, the liver itself, it cannot use ketone bodies. It just produces these ketone bodies for the muscle to use. And why can the liver use these ketone bodies? Because there's a certain enzyme that breaks it down that it doesn't have. This enzyme is thiophorase. Okay. So once these ketone bodies form, they go to the muscle, the brain, and then they utilize, they break down to produce ATP for energy. And then lastly, we have lipolysis. Okay, next, the muscle. The main energy source would be fatty acids. However, would also have a partial shift into using ketones as an energy source. Why would that happen? Let's see why that happens. Okay, now we said in a prolonged starvation state, you have gluconeogenesis, the liver produces, there's a lot of gluconeogenesis, right? And this means protein, a lot of proteins getting broken down to amino acids. Now this in long term wouldn't exactly be good because we want to preserve protein. We need protein for other processes. So to prevent this, to reduce the protein breakdown, the muscle starts using ketone instead to produce energy. This whole thing is called protein sparing. Okay. Lastly, adipose tissue. Again, the main energy source is fatty acids that we get from adipose tissue. No, fatty acids, yeah, from fats. And this is formed by lipolysis. The enzyme here, again, is the same one, hormone-sensitive lipase. And just repeating the whole thing, it breaks down triacylglycerol to fatty acids, which enters the Krebs cycle, gives energy, and then the other fatty acids moves to muscles and liver to produce energy. Okay, that is it for the three phases. Does anyone have any questions? Okay. And just to recap, we have three phases. Let's start with the feeding stage, well-fed stage. And here we know the brain RBCs, they use glucose as the main energy source. Liver uses oxidation of excess amino acids, and everything else, the muscle and adipose tissue, they use glucose as the main energy food. And the next stage, the early fast stage, we have, again, RBCs, insulin, they use glucose again. Now, do you guys remember with liver, muscles, and adipose tissue, they have one thing that they utilize? Fatty acids, yes. Lastly, starvation stage, RBCs remains the same. What does brain use? Ketones. What does liver use? Fatty acids. And what does the muscle use? Fatty acids, yes, and ketones. Okay. Okay, and lastly, this graph, it's and it basically shows how glucose is, how when you give glucose, how it gets used in different times or like throughout a time period. So in the beginning, when you ingest glucose, you'd have, and the body would start using it up. The muscles, the organs, liver, they'll all start using glucose for energy source and for storage. So you'd see that glucose starts decreasing. And then you'd also find glycogen increasing because glucose that you take in starts storing up. Excess glucose starts getting storing up. Okay. Then eventually, after 3 hours, after you're done with the feeding state, you're entering the early fast state, right? At this point, glucose is low, so glycogen starts breaking down, and you find that it starts decreasing. And then on the other hand, you'd find gluconeogenesis increasing. Why does that happen? Because glycogen stores start getting used up, and then we need glucose, so we get it from amino acids. And then eventually, after 24 hours, we enter the starvation stage. At this point, gluconeogenesis starts decreasing. Why does that happen? Now we know we have low glucose and we need glucose, but we still have low gluconeogenesis. Low gluconeogenesis means there's less amino acids, so there's less breakdown of protein. This whole thing happens because of something called protein sparing. Yep. Okay. That is it for the lecture, guys. Does anyone have any questions? Thank you.